Good afternoon, everyone, and thank you for giving up your time to listen to my project introduction with the title Elucidating the Role of Compatible Solids in Tolerance to Oxidative Stress. Hopefully you guys enjoy this. My talk this afternoon will include the definition on terms oxidative stress and ROS. Then I'll be explaining the compatible solutes, their main functions, some examples, and looking at the positive negative effects of compatible solutes when applied directly onto growing plants. After that, I'll be talking about my project and then the progression I've made so far, and finally I will end my talk with some relevant conclusion. Oxidative stress. In lame term, oxidative stress is any kind of stress that brought upon plants and caused damage to them. But smart people would define it as the physiological conditions created from severe and long-term redox imbalances due to the lack of electron as a direct impact of stress, causing cell injuries and triggering the signals and defense reaction in plants. And when the stress is combined with activation of oxygen, or shall I say overexcited due to the stress, we would have ROS, which are the byproduct of normal metabolic processes, which are respiration and photosynthesis, primarily responsible for the impairment of cellular function under numerous stress conditions. Some of the well-known ROS are singlet oxygen, superoxide radical, hydrogen peroxide, the popular one, hydroxy radicals, the nasty one, and also singlet oxygen. What do they do? As mentioned before, ROS are produced continuously as products of various metabolic pathways. Even when plants are growing under non-stress conditions, in normal days, these ROS are scavenged by a variety of antioxidant defense systems that prevent them from reaching that toxic level. But when present excessively, when the balance between production and elimination of ROS is disturbed due to stress like water loss, salinity, drought, etc., it will disrupt cell function, which induce cell damage by inducing the degradation of pigments, proteins, lipids, the nucleic acids, which ultimately result in cell death. Now, the implications towards plants are obvious in the form of physical damage like turgor loss, necrotic stunted growth, and reduce in yield. So how do plants run away from this kind of situation? They can't. Therefore, they have to either die or to adapt to their given environment. This is why we need to talk more about the compatible solid as plants' protection mechanism among many others in order to cope with stress. Compatible solid, what are they? Compatible solids are small organic osmolytes that may accumulated in cells at a high concentration without affecting metabolic reaction in either the cytosol or major organelles. What are they? They can be in the form of amino acids like proline, polyols like mannitol, sorbitol, non-reducing sugars like sucrose and trehalose, quaternary ammonium compounds like glycine betaine, some polyamines, and tertiary sulfonium compounds. And what do they do? Let's look at the proposed function of compatible solids. Now, we previously thought that compatible solid has a lot to do with osmoregulation. That is, the increase in cellular osmolarity would lead to influx of water and in the same time reducing the efflux of water. But we soon learned that potassium together with sodium and chloride in its adjustment has a lot to do with stabilizing the cell turgor because of the vast amount it produces during stress. Therefore, this conventional concept is no longer applied due to the mere reason they only exist in a very low concentration and they're very expensive and slow to make. Therefore, the opinion on their direct role in osmotic adjustment in plants is no longer relevant. However, the plant physiologists around the world strongly concur with the suggestions that because compatible solutes are mostly hydrophilic, they could replace water at the surface of protein, thus acting as osmoprotectants. Important in membrane protection, act as chaperones, oxygen radical scavengers, which is the prime suspect in my work, as well as many other functions as listed here. In saying so, we are still in the journey of discovering the specific roles of each compatible solute have, and how do they do it in intracellular level, which ended up helping the stress plants. Here is another list of documented roles in some compatible solutes with references, which are mainly explaining their in vitro roles. But how about in practical? Sorry, I'm gonna click this. Now, according to the recent findings, it is concluded that creating transgenic plant species specified in producing high organic solutes by gene selection not only counterproductive towards yield, but they're also apparently unable to produce sufficient amount of targeted solutes to ease the stress. However, the idea of applying the compatible solute exogenously onto plants look rather promising, but not without consequences. These are a few collection of documented findings explaining the roles of osmolytes when applied exogenously. Those one in blues, work and conclusion made by Sergey. I thought I better have it up there. Anyway, the major conclusion that we can make out of this little exercise is the fact that plants only need a low concentration of osmolytes for it to work properly. Moderate or higher amount was found to be detrimental for plants, which I thought justified the fact that plants can only produce a little amount of compatible solutes. Let's talk about the project, the fun part about this. Let's talk about the experiment, the fun part about the project. The topic is still elucidating the role of compatible solids and tolerance to oxidative stress. Our current aim is to quantify and to investigate the presence of compatible solids in plant across species, between dicot, monocot, and also between halophyte and glycophyte. We targeted 7 plant species, which are barley, maize, sugar beet, pea, prairie grass, Puccinella, and R. triplex. We treated them with salt in the range of 50 to 500 millimolar, as well as waterlogged treatment. And we harvest the young, mature, all leaves, also the whole plant for biomass. Project life cycle. I call it project life cycle because it has and still is a repetition exercise for me. I started sowing all the plant species in the same time. Let the plants grow until they reach a decent biomass, ranges around 16 to 45 days except for R. triplex, before I can put the treatment on them for 3 weeks. When I harvest them, I make some immediate measurements, prepare the sample so I can do the quantifying work on them, which only means I have to analyze them later. Now, these pictures are taken when I first started treatment for salt and waterlog. The first lot were barley, maize, and peas. Each plant species has 6 replicates, and each pot contains 3 individual plants. These pictures show the reaction of barley, maize, and peas under control, 100, 300, 500, and waterlog treatments. Soon after the harvest, I decided to add 2 extra treatments, 50 and 150 millimolar salt, so I can see the cascading effects on maize and peas. But then I thought I might as well do for the rest of the plant species too. Therefore, I have prairie grass, sugar beet, Puccinella. There will be more Puccinella coming to harvest for the 50 and 100 millimolar salt treatment. And of course, the R. triplex still have a long way to go because They are very, very slow growing. Data collection. Currently, these are the main information that is needed for my analysis. Plant height, above ground biomass, chlorophyll fluorescence, leaves osmolarity, certain potassium content, and the most important thing is quantification of compatible solutes using HPLC. I do face a few problems though, like finding the most convenient and suitable method or protocol on how to get a full extraction on plant material for HPLC. And the second one is I still need to talk to some people in Launceston on using their microwave digestion to get the total sodium and potassium content readings. And the last one is I still have issues with fungus gnat in the glass house. Conclusion. What's my conclusion? As a conclusion, the main message to take home here are osmoprotective and regulatory roles are the main functions of compatible solutes. Apparently, only little doses is needed for compatible solute to work properly. The excessive amount would only give detrimental or no positive effects on cells and or the plants. I didn't cover the next 2 points, but hopefully in more detail in my next talk, nonetheless worth mentioning. The first one is there may be more than one function for particular osmolytes, and different compatible solutes could have different functions. Now, point number 2, it is not yet known why different stress conditions should favor the accumulation of different osmolytes in members of one family. And the last point, to wrap up my talk for today, at this point almost no practical use been made of compatible solute in plant breeding, since we don't fully understand what they do, let alone how we can't even predict what uses they might have in near future. However, the large role played by compatible solutes in resistance to stress makes understanding them essential to any future work that focuses on the cell and also on the molecular level. There are many people to thank. I will have to thank Sergey and Lana for being good bosses. No, Davis, CSL, Caroline, Angela, Phil for their technical help. All my lab mates, especially Phan Rong. Where's Phan Rong? Oh, there you go. Phan Rong and Will for sharing the ideas and knowledge. To my dear husband, of course. Thank you for being a very good chaiwala. The last but not the least, all my intermediate families for their love and support. There are some references if you're interested.